Hello everyone and welcome to my channel Life Sciences. This is Dr. Ajni Sharma and today I have chosen the topic flow cytometer. Why I have chosen this topic? Because yesterday I got a question from my student. This was the question. So actually this is not as tough as it looks like, as I have discussed previously also one question that about the pedigree analysis. That was also very complicated. That seems to be very complicated, but actually that was not. And kindly ignore the background sound because nearby some plumber work is going on and I cannot work on that sound. So kindly ignore that sound. If I will concentrate on that, then I will not be able to record my class. Okay. So to discuss this, I will be discussing about the FACS. What actually FACS is? FACS is actually a shorting of the cells with the help of fluorescent light or fluorescent protein. So the question is like that: 4 different receptors via A, B, C, D binds to the same ligand X, and in order to determine when receptors has the highest ligand binding affinity, following experiments was carried out. It means to find out of this amongst these receptors A, B, C, D, who has a similar binding site or has all these 4 are the receptors and can bind to a similar type of ligand X, but with different affinity. To find out their affinity, an experiment was conducted in which what they did, they have tagged all these 4 receptors with green fluorescent proteins, and then they put all these 4 receptors in the media where the ligand X is present, and this ligand X was tagged with the red color fluorescent protein. Okay, now let's see what actually the FACS do. What is the property of the flow cytometer and how we use to analyze such types of things or to study such types of things. Actually, flow cytometer contains a nozzle where it allows the passes of only one cell at a time, and these passes of one cell at a time will interact with a laser light. And what will happen, as you will go to the bank and you might be seen that they use some machine to count the notes. So you might have seen each time one note was passing out there also somewhat same techniques is used, where the passes of one note from a path. What will be done, suppose as here it is a laser light, and what will happen when any cell will pass or come on the way to the laser light, what will happen, it will stop the passage of light across itself. And what it will do, because of the stoppage of light, what will happen, some sort of signals will be missing from there. So just like that in the bank also, when one note is been passing from a certain path, the light got deflected, or the light or the connection between the light and detector will be detached, and they on that way they count the nodes. Similarly here also in FACS also, cells while passing through the laser light, what will happen, it will stop the passage of the light across itself, and it will cause the deflection of the light. Either it will cause the forward deflection, and the forward deflection is directly proportional to the size of the cell. Then what will happen, what it was done, depending on the detection or whatever they detect because of this light or deflected light, which will be detected by a detector here. This is a detector. This detector, what it will do, it will detect the deflection or forward deflection or the forward scattering of the light, and depending on this it will plot a graph between the voltage and time, that after how many time and how many, or you can say it will particularly analyze the number of cells with respect to their size. Now what happens, the peaks particularly decide. Here we can see 2 peaks are there. One peak is more higher than that of the other peak. The higher the peak means that larger the size of the cell is. So first thing it do, along with the forward scattering, the FACS or flow cytometer analyze the size of the cell. Now we know that when any particle is been exposed to the light, they will deflect the light with different angles. So about the forward scattering, we came to know that cells size of the cells will be analyzed. And what about the sidewise scattering? Because of the sidewise scattering, what will happen? The sidewise scattering will also plot a graph similar to that of the forward scattering, but only thing is that instead of analyzing the size here, what will be analyzed here? The shape and complexity. Shape and complexity will be analyzed instead of the size of the cell. Now, what happens when using this sidewise scattering and forward scattering? What scientists analyze or researcher analyze? The researcher will analyze the size of the cell. Suppose they got a dots, because as just like the Ramachandran plot, also here we the result or the whatever we detect will become in the form of the dots. So here, if the dots lies in this quadrant, then what does it mean? It means the size of the cell is small and it is less complex in nature. If the dot lies in this quadrant, it means the cell is big. Here the cell was small, but here it is big with less complexity. Okay. But as we go up to the x-axis, what will happen? It will show that the cells or the content of the cell is much more complex. Here the cells are more complex, but it is of a small size. And this side the cells are big with more complex in nature. So with the help of this FACS, we have analyzed one is that the size of the cell, other is that its shape and its complexity. Another thing which we can analyze using the FACS is that we can recognize different types of cells present in a group of cells. Wow, we can do that. Suppose we have a cell having a certain marker over this, like some triangular type of markers are there, and there are another cells which are having some oval type of markers are there. Now what we will be going to do, we will adopt two different antibodies which will be tagged with a fluorescent compound. Suppose this has been tagged with the green fluorescent compound protein, and this antibody is been tagged with the red fluorescent protein. What will happen? Depending on their specificity of binding of the antibody, both of them will bind to a specific marker. Okay, here we have two cells, one with a triangular marker, another with the oval marker. There may be some cells may be present where it may have both the types of marker, both triangular type and both the oval type, or there may be some cells which do not have any marker. Now with the help of FACS, what we can do, we can even sort out these different cells altogether. Before that, I am focusing, as per the question, I am focusing on the FACS. I will explain that also, how we can sort such types of cells. So before that, let's start with this thing, that we have what we have done. We are having two types of cells, one with the marker having some triangular in shape and other with the oval in shape. When both of these been tagged with different antibodies with different fluorescent color, then what will happen? We will have a two detector will be there. Actually, in fact, 18 different colors can be detected, or 18 different marker can be detected at a time. So here we have used two, so I have shown only two colors deflection. So what will happen? Because of the sidewise deflecting, one detector will be detecting the red fluorescent color, and other will be detecting the green fluorescent color. And this is the monochromator which is lying over here, which allows the passes of a certain wavelength of light, and this will deflect it toward the detector. And this detector will again analyze the data in the form of plots or histogram, which we can understand, because we cannot understand how much they are absorbing, and what or they can simply give us a quadrant plot like structure, from where we will be analyzing the data, that which type of proteins are present over here. Now we have tagged both of the cells, one with the green fluorescence and one with the red fluorescence. Dots which are present here will indicate that simple things. Put here 0 So we know that if we will go toward the right, we will having a positive value. If we will go toward the left side, we will have a negative value. Similarly, when we go toward the down, we will have a negative value, and when we go up to the axis, we will have a positive value. Simply, just like we have analyzed in the Ramachandran plot, minus sign will be here. It means this area or this plot will be containing a cell which will not having any types of marker in that, which I have shown by the blue cell. Now come to the next. This is the cell where it will be showing the dot with the help of dotted plot or with the help of histogram, whatever types of analyzer you have. It will show those types of cells which are being tagged with the green fluorescent color. It means, but here it is what? It is below the 0 along with the y-axis. So what it is, here we have red colored tagged cells will be negative, while the green color will be color tagged protein will be giving the positive result. It means highest amount of green fluorescent tagged protein will be here. Moving to the upper side, here in the upper side, what we will be having? We will be having more number of cells with the protein tagged with the red fluorescent color protein. We will be having a higher concentration of cells over here, while the green fluorescent will be almost negative, while will be very less. Sorry. While in the uppermost quadrant, what we are having? We are having a mixture of the red as well as green color tagged antibodies or the cells which are having both triangular as well as oval marker over on its surface. Now, only this thing you have to remember to analyze the questions which was shown above. Now, coming to the question. What was the question? Question was that both I better use the color. First, the ligand is tagged with the red fluorescent protein, and what about the receptors? Receptor is being tagged with the both all the 4 A, B, C, D. All the 4 are being tagged with the green fluorescent protein. So how we can analyze that they are binding with the red color fluorescent protein or not? Moving to the back to our slide. They will be lying on the region, this quadrant, where both red or green fluorescent protein will be in higher amount. So moving to the question, just put the quadrant where it is having the highest number of value here. This is receptor number C. Why it is so? We can say, you see here only 5% of green and red fluorescent proteins are here. Okay. And after that, when this protein has been shifted to the because what we will be getting, the receptor which is having more affinity toward this X ligand which is been tagged with the red fluorescent protein, then what we will be having, this green receptor will be tagged with this red color. So where we will get this data, or where the deflection will be more? It will be definitely in the area where it is showing positive for the green and positive for the red or the red fluorescent protein, both. Once again, I am simplifying it. There may be confusion between receptor B and C, because initially I was also confused. What it is there? It is 52, which says that here unbounded X protein is there. I better the quadrant here says that here only X ligand will be here. Why? Because it is been tagged with the red color protein. Here also X red colored protein will be there, but in this quadrant red color protein or the ligand will be also there, and the green color tagged receptors will also be there. Okay, where here in this quadrant only green color or the receptors will be here. Here also only receptors will be here. Okay, while in the upper quadrant, in this quadrant, receptor has bonded to the ligand. It means the receptor which was red in which was tagged with the green fluorescent protein has now binded to the red fluorescent protein, and the concentration of both the protein should be high, green color as well as the red color. So moving forward, why I was confused, I was just looking at this value that here only X ligand is high. It means unbounded X is higher. The percentage of unbounded ligand is high. It means the receptor has not bonded to 50% of the 52% of the X protein or X ligand, whereas here 52% of the X ligand is been bonded by the receptor. We can analyze like this, because here both the receptor as well as ligand concentration are high. Understood? Now once again I have told you what that we can separate even different types of cells. So here we have taken two types of cells with the two different marker here, one with the triangular marker and one with the oval marker. Now what we will be going to do, we will tag both of this with a fluorescent compound or fluorescent protein just like before, along with the charge also. Suppose I have tagged the green fluorescent protein with the negative charge and red fluorescent protein with the positive charge. This can be done in two ways. Either I will tag the charges directly on the fluorescent protein, or I can stimulate the vibration to the cells, and the vibration will be like that the cells which will be tagged with the fluorescent protein will be positively charged, or the cells which will not be tagged with the fluorescent protein will be negatively charged likewise. Now what will happen, they will bind to their respective antigens present on the cells. Now what will happen, we know that the negative charge will attract the positive charged cells. So we were having the what we were having, we were having red color with the positive and yellow color with the negative charge. Now here then what will happen, red color being positive charge will come and will be deflected toward the negative electrodes present over here, and the green color will be deflected toward the positive color side, and the cells which do not have any types of charges or having both the charges will be go straight toward the other container or other test tube which we have put here. So to sort out the cells as per their proteins present on the surface, what we can do, along with this all the setup of the fluorescent flow cytometer, we will put another additional electrodes over here which will deflect the protein or the charged protein, and depending on the charges over the surface. This can be done artificially, or we will induce a vibrational charges over the surface also. So this was all about the FACS. So once again I am summarizing it. What is about the FACS? FACS is containing a flow chamber where we put different types of cell suspension. It is designed in such a manner that at a time only one cell will come out of this nozzle, and when it will come down and it will interact with the laser light, then what will happen, we know that any compound which will come in on the way to the light, it will deflect light, some it will be absorbed. So the forward deflected light will be detected by the forward analyzer or detector, and here each detector will be having a band pass filter which will allow the pass of a certain wavelength. And here you can see the sidewise scattering or sidewise scattered light will be deflected toward the detector which are present 90 degree toward to that of the angle of laser light, and this deflected light will be detected by the side scatterer analyzer, and it depending on their analysis we will be having a certain type of graph which we have discussed now, and there will be diachromic mirror also which will allow to define the specific type of wavelength to be detected by the detector, and detector will show us the graph in the form of some histogram along the x and y axis which we have discussed. And what will happen, depending on the charges we have implemented, we can also sort out the cells. So with the help of FACS we can analyze the what we can analyze the size, shape, complexity, complexity of the cell, as well as we can sort out or the cell on the basis of the charge, negative or positive, or it can be neutral. So we can do all these things with the help of FACS. So these were about the FACS. We have discussed one question of the CSIR JRF net also. Don't forget to subscribe and share my channel, because I will be uploading similar types of videos, many more. And I have just now, before this video I have uploaded video about the thermodynamics questions which was asked in previous year CSIR JRF net. I given the link of that in my description box.